Hi everyone, this is the continuation of Module 4. This is Module 4B of the 19th Century Transformation of Western Medicine. So back to Pasteur and the two other crucial discoveries that he made, which were that specific bacteria cause specific microbial infections and the development of experimental immunology. The first specific bacteria that Pasteur isolated were bacteria that infect silkworms. The production of silk was a major industry in France, so this was a very important discovery. Pasteur identified two specific bacteria that each caused a specific disease in silkworms. They were called pébrine and flacherie. Once Pasteur had proven this through painstaking research and trials, he surmised that it must be a similar condition that caused illness in animals and humans. In other words, specific diseases in animals and humans are also caused by specific bacterial or microbial infections, just as they are in the silkworm. So, with the idea of specific infections in mind, Pasteur turned his attention from silkworms to farmyard diseases, specifically chicken cholera and anthrax, which we'll talk about later, which is a disease that kills livestock. Pasteur realized that he could isolate microbes and then inject them into healthy specimens, and then those healthy specimens became infected with the microbe that he had injected into them. In other words, he showed the process of infection passing from one host to another. But his vision went beyond simply demonstrating the germ theory, although he did that. In addition, he developed the public health practice of widespread vaccination for a variety of diseases, and thus he helped found the discipline of experimental immunology. Now, a bit of historical reference. The idea of vaccines had been known for perhaps hundreds of years before Pasteur's experiments. There's evidence that the Chinese and the Arabs used vaccines for smallpox dating back to the 16th century at least, maybe long before that. The idea of smallpox vaccination was popularized in the West by the English physician Edward Jenner in 1796. The basic idea is that Jenner realized that cowpox confers immunity for human smallpox. So, if you scratch someone's skin and rub some scabs from a cow's pox infection into it, that person gets an immunity to the smallpox that infects humans. Alternatively, you could rub smallpox scab from a human into a scratch onto another human, which could give them, which could also confer immunity, but is more dangerous. The key point is that cowpox does not make a person get sick, other than maybe a few blisters. But it does confer immunity to human smallpox, which is a very deadly disease. So, vaccination, or the introduction into the body of either the whole or part of a disease causing microorganism in order to teach the immune system to attack that organism, had been known for some time. Of course, we understand now that the vaccine primes the immune system to produce antibodies or teaches immune cells to recognize and attack the organism. The problem, of course, was and remains how to stimulate the immune system without causing the disease itself. Jenner benefited from the crossover immunity that exposure to cowpox also confers immunity to human smallpox. He realized that humans don't get sick from cowpox, but they do get immunity to smallpox from it. But Pasteur did something else, something totally new. He used the concept of attenuation, or weakening microbes in order to safely confer immunity when introducing harmful microbes to the body. So, what is attenuation? Attenuation is the idea that live pathogens can be introduced into the body, but only after they've been weakened in some way so that their virulence is diminished. So you could weaken the pathogen by heating it or by exposing it to some chemical. Then this weakened pathogen is injected into the body. Pasteur realized that a weakened pathogen will not make the patient sick, but it will give them immunity from further infections by that pathogen. Now, this discovery of Pasteur's of attenuation was made by complete accident during his work with the bacterium that causes chicken cholera. Pasteur had isolated some chicken cholera bacteria and was in the process of injecting it into healthy chickens in order to study the rates of infection. He prepared several batches of chicken broth that he grew the chicken cholera in. Now, here's the accident part. Feeling overworked, Pasteur decided to leave his lab for a few weeks of vacation. When he departed, he left several batches of chicken broth that were full of bacteria on the shelf in his hot and stuffy lab. When Pasteur returned from his vacation a few weeks later, he found initially to his frustration that when he injected this culture into chickens, he wasn't able to infect them with cholera. So he got a fresh batch of bacterium and injected the same chickens with it, as well as a lot of new chickens. And he made a surprising discovery that the original chickens that he had injected with the old vaccine that had been exposed to heat, they remained healthy when they were injected with the live new bacteria. In other words, they were immune, while the previously untreated chickens sickened and died. Pasteur repeated the experiment several times with the same results and concluded that the summer heat had somehow attenuated or weakened the culture of the bacterium, and that this weakened bacteria conferred immunity when injected into other chickens. Pasteur had proven accidentally that heat could kill or weaken samples of chicken cholera to the point that they no longer caused death but still induced immunity. Having discovered attenuation with chicken cholera, Pasteur applied the same principle to the different disease of anthrax, a bacillus that had recently been discovered to infect livestock. In Pasteur's famous anthrax experiments, Pasteur took two groups of healthy sheep. Uh, in the first group of 24 healthy sheep, he injected them with live anthrax, and they all died. In the second group, Pasteur first injected these 24 healthy sheep with an anthrax that had been chemically weakened in his laboratory. All of these sheep lived. So he took these same 24 sheep that had been injected with this chemically weakened anthrax and then injected them with a live anthrax. He also injected 24 more sheep with live anthrax. The ones that had been just injected with live anthrax all died, but the 24 that had been pre-treated with the chemically weakened anthrax, they all lived. So when they were re-exposed to live anthrax, they all remained healthy. In other words, he had shown that he could confer immunity to anthrax using a chemically attenuated anthrax bacillus. So now Pasteur had shown that he could confer immunity for chickens with cholera and for livestock with anthrax, but he hadn't yet conferred immunity in a human being. And the very first experiments that he did that showed that he could confer immunity in a human being through a vaccination was with the experiments that he did on rabies. Now, rabies is not a super common disease, but at the time it was a very fearful disease. People were very frightened of getting rabies because it was a certain death, and it was a horrific death. Um, so, rabies, as we know, is not a bacterial disease, but a disease caused by a virus. Now, how Pasteur went about these experiments was he first isolated uh, rabies in a fox, and he took cerebrospinal fluid from the fox and he injected it into rabbits. And then he would wait until the rabbit died, and then he would take out their spinal columns, and he would allow them to dry in the air for between 5 and 10 days. And then he would make a fluid off of the dried spinal column and inject it into the next rabbit. And he would wait until that rabbit died, and he would do the same. So he would pass the virus through a series of rabbits. At the end of that process, he took the serum that he developed off of the spinal column of the rabbit and he injected it back into a healthy fox. Um, the healthy fox did not get rabies. And in fact, when the healthy fox was injected with rabies, it still couldn't get rabies. In other words, he had shown that he could make immunity to rabies in a fox by attenuating the virus by passing it through an animal of a different species, which was an enormous discovery. So, if Pasteur could create immunity to rabies in a fox by injecting it with a weakened virus that had been attenuated by passing that virus through a series of rabbits, could the same thing be done for a human being? Well, the Great Human Trial occurred in July 1885 with the famous case of a little 9-year-old boy named Joseph Meister who had been severely attacked and mauled by a rabid dog. And everyone was certain that he was going to die an agonizing death. So they called Pasteur because they had heard about these experiments that he had done in creating immunity in foxes. So Pasteur knew that rabies has a very long incubation period in human beings. So during this time period, he began vaccinating the boy with a series of shots of attenuated rabies viruses that had been passed through the rabbits, the exact same injections that he had been giving to the foxes that conferred immunity for them. And to everyone's amazement, Joseph Meister survived and became quite a celebrity as the first person to have ever survived after being severely bitten by a rabid animal. So Pasteur proved that you could inject humans with a virus that had been attenuated by passing it through an animal of another species, then injecting that into human beings, you could confer immunity in the human being. This was an amazing discovery. All of this fame generated by Pasteur led to the founding of the Pasteur Institute in 1887, with Louis Pasteur himself as its first director. The Institute followed the public health strategy of vaccination, pioneered by Jenner and now perfected by Pasteur. The founding of this institute gives us the first example of what would become a key component of 20th century science, which was the international scientific competition for national prestige. So much of what we've talked about up to this point has been the importance of science and medicine that began in the early 19th century in Paris, and centered very much on France. In many ways, the founding of the Pasteur Institute was really an attempt to recapture the glory of these earlier days of the Paris School of Medicine, because by now, in the late 19th century, most of the energy of scientific discovery had shifted away from France and towards the new laboratories in Prussia, or what we would call Germany. So, in our next module, we're going to start exploring the laboratories of Robert Koch and the rise of the new German school of laboratory science and medicine.